Dr. Habib Bokhari, Vice Chancellor, Kawsar University, Murray.
Bismillah ar-Rahman ar-Rahim. Assalamu alaikum, everyone. And I hope you can hear me loud and clear. So let me quickly go through. My topic will be slightly different what other presenters basically were presenting this day. I'm going to basically talk about how we can take help from microbes in improving our basically lifestyle. So there'll be quite a few stories. I'm going to tell you about 3 or 4 stories basically which originated from my lab work in order to give you a idea that how these microbes can be used as factories to produce various products which can be utilized for the public health. So I'll start with few basic definitions for the students. Basically, you need to understand what is the difference between basic science in the laboratory, what is the difference in applied science in the laboratory, what's the difference basic science in the clinical matters, and what is the difference between applied science in the clinical matters. So you can apply all those applications in these areas by understanding the difference between them. So as you are all aware of the fact, basic science basically seeks different answers of different questions and create a fundamental knowledge in area of physical or biological domain. Whereas the applied science in laboratory studies is directed towards the specific issues and problems. And similarly, basic science in clinic seeks fundamental knowledge in various pathophysiological issues. And applied science in clinic is basically leads to a translational research with the development of medicinal interventions in controlling various diseases. So I'll just give you a simple background of these definitions. Now we'll move on to the how basically we talked about the translational research. So this is where basically the world is moving towards, and Pakistan especially moving towards applied area, and a lot of funding and opportunities are available in this area because prior to that there was a focus on a basic research mostly, but since now the shift of the government and the policy makers is towards the applied area, applied research, a lot of funding opportunities are coming in this area, and I'll talk about those quite a few, 2 or 3 case studies where basically you need to learn how these applications can be made in these areas and how there is science and research activity can be conducted in applied areas. So there can be various ways. It's not only the beneficial microbes, but how we can take advantage of a microbe. So there can be 2 ways of dealing. The idea or a theme of this workshop is purely on a beneficial microbes with reference to genome structures and genome sequence structure-function relationship. But I'll also give another taste that how these microbes can be used in order to get the commercial benefits. So that's there can be two ways. There can be 2 kind of a case studies: directly the beneficial microbes and how the microbes can be utilized in producing various products which can be commercialized. So just giving you a background that what is you are all familiar with the fact that what is the metagenomics and how you can basically utilize metagenomics in discovery process. So there are 2 definitions where the metagenomics approaches can be explained very well. One is the function-driven method, where the metagenomic libraries are initially screened for an express trait of which we are interested in basically, and then there's a sequence-driven method where the libraries are initially screened for particular DNA sequences. So both have their own advantages and limitations. So what is the advantage of a function, limitation of a function-driven method? Basically, it requires the expression of a function of interest in a host cell. And whereas what is the advantages of the sequence-driven method? That is to give a picture of a genetic biodiversity which is present in a given environment, and it is basically also provide you insight of the functional diversity of the whole genome content which has been isolated from a certain environment. So this is how basically they differentiate. Now I'll give you one classical example of a biotechnology industry and how it grows initially with reference to our therapeutic molecules. You are all familiar with the fact of insulin story. I'll just refresh your minds that it started in 1869 when the pancreatic islets were originally discovered, and then the molecule which was commercialized came in 1980s, genetically engineered microbes, genetic engineered insulin was basically available. So that was the initial era where a lot of, it took almost a century in order to come up with a therapeutic molecules in the commercially available for the treatment of diabetes. But with the discovery of a metagenomics, now on my right side, if you can see that this metagenomics, as I already explained, function-driven, sequence-driven approaches, these discovery process has been basically now much quicker and faster. And I'll go and talk about briefly about this, how this discovery can be made, and where the microbes can be also helpful in terms of producing those molecules and their role in basically taking full advantage of these discoveries for a commercial purpose. Now, if we look at the protein discovery process, we all know that the functional molecules are actually the proteins, which are the backbone of the biotechnology industry. And we all know that there are certain processes which are involved in this discovery process, right from the cloning, expression, purification, and then understanding the function of the protein and making them a commercialized molecule. So this is a complete cycle I've just given you in a background. Now, if you look at these, now this whole concept we applied back in 2005 in identifying some novel fluorescent proteins, which we also, these are the commercial molecules isolated from the, originally isolated and reported from a jellyfish Aquaria Victoria, and the three individuals shared a Nobel Prize on that back in 60s. Now, what we did basically, I just go through that. I'll just give you a little background how these fluorescent molecules work. Basically, there are different wavelengths these different colors basically emit, and that because of that emission they are identified as blue, green, yellow, orange, and red. These are the commonly known commercially available fluorescent proteins, but we historically call them a green fluorescent protein, but actually it's their emission spectra which determines the color range. So on the shorter side you can see the blue with the 459 nanometer, and the longer side you can see the red with the 700 nanometers. And beyond that, no ultraviolet and infrared, nothing is visible to a common eye. So that's why they are not used in fluorescent tagging or in a molecular. So what basically these fluorescent proteins, what is their use? And you are all familiar with the fact that they are the most popular molecules which came into existence after that 1960s when it was discovered. In every molecular microbiology or cellular biology lab, they can be used in visualizing those molecules or cellular organelles which were initially translucent or we were not able to tag them, or we were not able to visualize them properly because there was because of a black and white kind of a microscopy which was available till the time of 1600, when the Hooke was the one who basically identified and started looking at under these cellular organisms under the microscope. But now they have become more colorful, and you can tag various functions within a cell by tagging them with various colors. So that's what the importance of this molecule is, that, and these commercially available molecules like Clone Tech basically sells them for one single protein. You can get that 100,000 rupees. So that's very expensive molecule, and that's why we have to work on locally identified molecules which can be commercially made available to the scientists, so they can be cost effective and easily available. So these are the different diagrams and showing that these have been basically used in making various ornamental plants, and has been used in understanding different diseases and making different tissues colorful, and can understand the mechanisms going on inside the cell very clearly, minutely. Now, what I'm going to talk about that how we basically discovery process involves in identifying these molecules is a very simple process. You normally take these tissues. I worked on a this is basically a horn coral. We call it a basically a Hydnophora rigida, which is a horn coral, have been isolated from a Pacific Ocean while I was in USA doing some working as a research scientist. And the simple process basically involved in cloning these cDNA molecules and identifying these molecules as a green and cyan fluorescent protein based on the fact that you just clone them, and these are the proteins which show their presence by their visible or phenotype or a trait. Or there was a problem on my side.
Welcome back, Dr. Habib.
Yes, I'm gonna finish that and wind up quickly. So I was mentioning about the discovery process of these fluorescent molecules. So it was basically, now it was, we selected these fluorescent molecules in a vector on E. coli, and a couple of strains which we isolated were found to emit green fluorescent and cyan fluorescent proteins. And those were the discoveries which basically took about 6 months to identify these molecules in screening. And now they have been characterized through various tools in order to find their spectrum, fluorescent spectrum. And this is how they look like when they were expressed in various bacterial strains. Now they're ready to be commercialized. Now bacteria has been the tool. What I'm going to say that here, the bacteria has been a tool in a discovery process in identifying novel molecules from the eukaryotic region and quickly expressing them in a bacteria and screening them. And now these molecules can be now are in a process of commercialization where there a lot of protein engineering can be involved in making them more fluorescent. So that's what we have been working, and we have identified that these fluorescent molecules were the shortest and smallest tag, and they were the novel proteins and were different from any other proteins which have been reported so far. So they have their own potential as well. So they can also be utilized in biosensing reporters as well. Now, this is another beneficial use of these microbes. When combined with these fluorescent molecules, they can be used to detect, for example, pesticide in water. Now you can see the color change here. Whenever they bind to these pesticides, methyl parathion is an example here. So there the color quenching takes place and they can be easily and quickly detected in a water. So now you can see that where it is basically degraded protein by binding the methyl parathion, it gives you a clear indication that the methyl parathion is present in the water. So pesticides can be detected because they have certain motifs to which these pesticides interact and denature the proteins. Whereas the color one, the number one is basically showing a fluorescent protein in a normal way, and number two is where it reacted with the methyl parathion. So this is the bacteria expressing these fluorescent proteins. So this is the structure of a fluorophore. I won't go into detail of this fluorophore, but they can also be used in detecting various metals as well. For example, there is a big problem in Pakistan of arsenic. Now, if you look at that, the last three slide where these nitrocellulose chips have been developed to which these bacterial cells were immobilized, and you can see a clear difference where they were treated with arsenic, the color quenching takes place, and they tell us that the arsenic is present where there's a normal control. And similarly, copper and other metals have been detected using these bacterial cells and immobilizing them on chips. Now, the other thing, now this is how this sol gel immobilization worked with reference to the E. coli. The other story which I like to share quickly is isolating certain industrially important enzymes from the cyclophilic microbes, which we isolated from glaciers, such as Pasu back in about 5 years ago. And we isolated certain molecules which have a commercial value to be used in various industries. So these organisms have the potential to sort of thrive or live under minus 40 degrees centigrade. So they have, the nature has kept a certain metabolic pathways active in these microbes, which can be exploited and used in various industrial processes as well. So as I just given you a brief summary here, that these microbial enzymes can be used in various industry, right from the leather, food, leather tanning and protein hydrolysis industry. And on the right side, you can see the agriculture sector, food waste degradation, and many other areas have been touched upon here. So these enzymes can be used. And what we did basically in this discovery process, that we identified certain molecules which were associated with our organisms of interest, which was in this case, a Pseudomonas was one of the species which we worked on, where basically it was used to produce exopolysaccharides, which has a cryoprotectant role. I'll show you in the next slide what is a cryoprotectant. Basically, it basically preserves the cell from at low temperature, and extreme freezing impact can be basically tolerated because of the presence of the exopolysaccharides. So exopolysaccharides can be oscillated from a range of microbes, right from thermophilic to the psychrophilic. So they have different properties, but we use these psychrophilic exopolysaccharides for studying our various processes and seeing their functional role. And one of the simplest way, another thing which we also isolated was the proteases. The proteases have been isolated in the past as well from various bugs, but one of the proteases which we isolated and seen from these psychrophilics have a very high active activity, and they can be used in dehairing. We know the leather industry is a major sector in Pakistan, and removing the hair from the leather, sometimes if the process is not very safe, it can damage the leather, and leather sometimes, and then the leather becomes wasted, and that's the area where the enzymes can be utilized, and we have successfully removed these hair or proteins from using these proteases in order to use them in the leather industry. So this was one of the very good example of using cyclophilic proteases. The other activity, as I mentioned, exopolysaccharides can be used as a cryoprotectant. And you also are familiar with the fact that glycerol is normally used as cryoprotectant in industry, but here is we will use the exopolysaccharide, and they have also they basically the example I given you earlier, where the green fluorescent protein has been expressed in bacteria. If we store these bacteria at -20 or -40 degree with the glycerol, without glycerol, and a cryoprotector, which in this case is the EPS, you can see in this diagram that the one which has been given GFP and EPS is growing more rapidly because the cells are surviving longer in that. So that is a clear proof of doing and showing that EPS has a cryoprotectant role. And this is a very simple way of doing this experiment. We combine the power of green fluorescent protein present expressed in the E. coli in testing all those commercial molecules such as EPS in this case to show their ability or capability to preserve the cells. So this is one of the major problem in where these preservative, whether you have to store the cells of for a longer term at a room temperature at a -20 or -40 by using a cryoprotectant such as EPS. So EPS can be used as a good cryoprotectant as well. Now another area is basically therapeutic molecules production. Then the vaccines can also be produced as in a factory, a microbial factory in a very systematic manners. So I'll just give you an example how these. I'm just giving you one example of a glycoproteins. Glycoprotein is a next generation. Basically, 70% of the therapeutic molecules are the glycosylated molecules, which industry is basically producing in recent times, and they can be used and produced in a bacterial cell simply in E. coli by taking the whole glycosylation machinery from a Campylobacter jejuni, which is one of the gut pathogen where initially this machinery was discovered. Now this whole thing can be shifted to the E. coli, which is the normal bug which we normally use in a laboratory, in order to produce various glycan or sugar molecules, and then sugar molecules are automatically attached to the acceptor protein molecules to make the glycoproteins, and those glycoprotein has a potential to be used as a vaccine agent. So I'll just give you an example. This is a very simple procedure which has recently been used in producing quite a few vaccines. For example, Haemophilus influenza vaccine is coming out as a glycoconjugate vaccine, and this is the future. Basically, livestock vaccines as well as the human vaccines are going to be produced within the bacterial cells, where initially the modification process was not known, but with this translation of one glycosylation machinery of a bacteria to another bacteria, it is possible now. So we can easily do that in a factory manner. This is how a factory works. That you can see this is an E. coli cells expressing both plasmids with the sugars as well as the protein molecules, and these sugar and protein molecules are added by the transferases present in these bacteria in order to produce the glycoproteins, which can be easily harvested compared to the traditional process, which is very tedious process, which we call a click chemistry. So now this is much easier to do that and produce. And this is how basically these glycoproteins look like. If the one sugar is added, we call a monoglycosylated. There's two sugars, diglycosylated, triglycosylated, and tetraglycosylated. So their molecular weight increases with the addition of each sugar molecule to the protein. So they can be isolated, harvested, and used and tested in various vaccine trials. Now, the final thing which I like to add, because a previous speaker was mentioning about the microbiome, gut microbiome, and probiotics role, and that's very important thing which is also coming up and commercially a huge multi-billion industry. Now, I've started looking at microbiotes by looking at, first of all, how these microbiome or a normal gut flora is disturbed. Now, there are various pathogens which are present or prevalent in especially third world countries, which are due to the sanitation and hygiene issues, foodborne pathogens, waterborne pathogens, actually ended up in the gut of the child. When the child is at a vulnerable age or early age, if they are started prevailing in their gut, and in addition to that, the malnourishment is there, these both combined together causes the stunting, which is an intellectual disaster or basically a disability. Now, what is stunting? And in easy word, I would say that stunting is basically is a process due to which the physical growth of a child as well as their mental capability is compromised, and as a result of that, the stunting basically appears. And in Pakistan, there are 37% children are born with this condition. So that's a huge problem. Now, one way of dealing that problem is to improve our sanitation hygiene condition. The other way is to restore the gut microflora. Now, we understand why it is important to restore the gut microflora. If you look at the central point of my slide, there are about 20 enzymes which are produced in the gut by the human genome, whereas the residual microbiota living in the normal flora in that gut produces about 260 more enzymes, which it also includes, which also produce vitamins and antioxidants. Now, these are very essential for the growth of a child, and moreover, when the microbes such as Campylobacter, E. coli, and other Helicobacter pylori are present in the gut, they interfere in the absorption process of a food. About 10%, more than 10% of the nutrition pass away unused when these organisms interfere in the normal processes of a digestion. So you can imagine the burden and where the probiotics can come and play a role in regulating and improving this dysbiosis condition, which is very important nowadays in our country. And this is how some of the work which we did. You can see the stunting. These are the Pakistani children we basically studied during our recent studies, and all the measurements have been taken away. And this is how basically the government is also trying their best to improve the nutrition, but nutrition alone cannot be the solution until and unless sanitation hygiene conditions are not improved. So with these words, I like to end my presentation. And obviously, this is the probiotic industry. It's a huge industry. And then there's a vaccine and therapeutic industry. Veterinary vaccine industry is going to be one of the major industries with the population, ever-growing population. And similarly, the probiotic industry is ever-growing as well, keeping in view all those. One of the conditions I have already highlighted, but there are a number of conditions where these probiotics can be used to improve that health status of an individual. So thank you very much for listening and being patient to be with me for 30 minutes or so. And I like to end with that. Dr. Shakra, thank you very much for inviting me. I wish and hope that I should have been physically there, but I was engaged somewhere else. So I tried to make this, a quick talk for my audience and your audience, and hope they have learned from this. And if there's any question I'd like to answer, they have perhaps 4 to 5 minutes if you wanted to ask any question.
Sir, there is a question in the chat box. Can you please answer that?
Oh, where is the chat box? If you can you read it for me? I can answer that.
Sure, sir. You have experimented this in vitro, but how about in vivo or in situ? Because this showed zone of inhibition on plates, but the atmosphere outside is little bit different.
So the question I like to reframe that the protease, you're talking about the proteases assay?
Yes, sir.
So what normally we do in a laboratory, we just carry out, this is a proteases assay, we normally carry out them with the milk proteins, right? The milk proteins, which are the casein molecules, are, basically, there's a litmus test of using them for understanding or studying the activity of the proteases in a simple way. So zone of inhibition, you can see that how it's clear the zone and eat away those proteases molecules. Those protein molecules or casein molecules determine the activity of the proteases. And then we normally use that assay that applies and purified those extracted proteases on the skin of the leather. And then we have taken away all those hair and removed those hair non-invasively without damaging the skin of the leather. So that was the simple proof of principle, testing their activity initially in the lab, then applying that on the actual material so that it shows that it has a potential to improve that. So that's something which I understand. Yumbi, I have been able to answer your question.
Thank you so much. It was really informative, sir.
Thank you very much.
Dr. Habib, thank you very much. It was an honor having you.